Technical limits of mass treatment.
As diagnostic techniques improved, so did the availability of drugs for mass administration. The various medications introduced after Korean War enabled people to actively fight roundworm infections. But they were not without side effects. Until the 1950s, the main antihelminthics used were santonin and seaweed called Degenia. Degenia is one of the most common seaweeds found worldwide, including Korea and Japan. It grows on rocks underwater. Boiling this seaweed can extract the active ingredient that are effective against roundworms. The fact that it can be made from common seaweed makes it relatively easy and inexpensive to obtain. However, it smells foul, can cause dizziness, and has side effects such as yellowed vision. The drug was not properly purified, but simply collected and boiled from seaweed, resulting in inconsistent concentrations and low antihelminthic efficacy. Both drugs had many side effects, including nausea and impaired vision, and were highly toxic, which resulted in infant and child death from overdose. Santonin was the drug of choice for mass medication until the early 1960s. It was extracted from certain wormwoods, which was first successfully extracted by the German pharmaceutical company Merck in 1830. Until the 1940s, the raw material for santonin used in East Asia was mainly produced in the Russian highlands. However, during the Cold War, the export of raw materials from the Eastern Bloc, the main producer, was halted and caused the price of the drug to rise. And this made it difficult to administer mass medication. The rising cost of raw materials also led to the circulation of fake medications that contained little of the active ingredients. In 1965, 20% of the 300 tablets purchased by Seoul to administer to the city's school children were deemed to be of inferior quality. The medicines were not delivered on time, so the dosing schedule had to be pushed back.
Technical limits of mass treatment.
In 1969, the first national mass deworming program was carried out and covered 3 million people in schools across the country by using a drug called pyrantel palmate. Developed by the American pharmaceutical company Pfizer, it was effective not only against roundworm but also against other intestinal parasites. It was the first broad-spectrum antihelminthic drug to be introduced into Korea. The problem was that there was no technology to produce the drug domestically, so the entire 5 tons of raw materials had to be imported. The Korean government didn't want the situation, since it was urgent to curb foreign currency outflow as part of its economic development. Rather than dosing all children, regardless of whether they were infected or not, Korea chose to selectively dose through mass screening, despite a high rate of parasite infection, in order to save money and foreign currency. With most of the raw materials for deworming pills imported and costing more than $1 million per year in 1974, non-selective dosing was a drain on the national treasury. In the 1960s, Korean pharmaceutical companies had limited capacity to synthesize raw materials, and most imported and processed raw materials from abroad. The customs clearance process was also very difficult due to concerns that pharmaceutical raw materials could be used for narcotics or adulterated medicine. By the late 1950s, 90% of imported pharmaceuticals were finished products and limited the import of raw materials. In the 1960s, Park Chung-hee's government restricted foreign exchange outflows to invest in other key industry. For the reason, the pharmaceutical industry, which relied primarily on imported raw material, faced a shortage of raw materials. When imports of raw pharmaceutical ingredients were stopped in 1966 to save foreign currency, the price of anti-helminthic drugs began to rise significantly. Periodic government embargoes and other restrictions on imports forced pharmaceutical companies to localize raw pharmaceutical ingredients as an essential part of stabilizing their operation. The same was true for companies that made anti-helminthics. As early as 1962, the Korean government implemented the Pharmaceutical Raw Material Protection Policy to promote localization of raw materials. The policy was providing a type of monopoly that prohibited the importation of foreign drugs with the same ingredient for drugs that were recognized as new synthesizing technologies in Korea. The government believed that the domestic pharmaceutical industry, which was small at the time, was unable to invest heavily in research and development and facility to localize raw materials.
Reverse brain drain.
Shinpung Pharmaceutical is a good example. The company that entered the pharmaceutical industry during this period by attempting to localize anti-helminthic drugs. Established in 1962, Shinpung's main product was anti-helminthic drugs. Until 1965, the company manufactured mainly from imported materials. The company then set out to develop a broad-spectrum anti-helminthic. In the 1970s, advanced anti-helminthic mebendazole was targeted to be localized. Developed by the Belgian pharmaceutical company Janssen in 1971, it was a true broad-spectrum anti-helminthic, effective against a wide range of intestinal parasites. It was effective not only against adult worms, but also against larvae or eggs. Until the 1960s, mebendazole was a breakthrough in that existing anti-helminthics were only effective against some parasites. However, Janssen's manufacturing method was expensive because the starting material was expensive, and the equipment to synthesize it was also expensive. Converting it into the intermediate required heating ammonia to over 125 degrees Celsius, which required specialized equipment that could withstand it. The high pressure and high temperature synthesis method also had a disadvantage of being unsuitable for mass production due to the high risk of accidents. A team of researchers at Shinpung Pharmaceutical spent a year in 1972 working to develop a new process for mebendazole on their own. But the technology and equipment available in the Korean pharmaceutical industry at the time made it difficult to follow Janssen's method. With little progress, in 1973, Shinpung Pharmaceutical approached the Korean Institute of Science and Technology, KIST. KIST was founded in 1966 with full governmental support and focused on providing technical support to industry. At the time, it was also implementing a program to stem the brain drain, bringing back talented Korean scientists and engineers from overseas. KIST offered full support returning scientists. One such chemist who returned to Korea against this policy backdrop was Kim Chung-sop, who specialized in chemically synthesizing drugs, especially in developing new processes for reverse engineering drugs. Previously having developed abroad, he was able to do this because Korea at that period only allowed patent for manufacturing method known as process patent. Unlike the current patent system, which grant patent right to the raw materials of drug, known as product patent. In other words, even if the same ingredient, mebendazole, is produced, if the manufacturing process is significantly different, it is recognized as a separate patent. Therefore, a key goal for Kim and Sinpung was to secure a separate mebendazole manufacturing process without infringing on Janssen's existing patent. In the same year, Kim succeeded in developing a new process for making mebendazole using inexpensive and readily available materials. He also utilized a method that allowed the reaction to take place at room temperature rather than at high temperature and pressure. And the catalyst for the reaction was common iron powder rather than an expensive precious metal. As a result, the overall cost of the process was significantly lowered. Despite the low cost of the raw materials, the yield was high. While the previous high temperature and pressure method only yielded 25% of the mebendazole from the raw materials, the newly developed method significantly increased the yield to 73%. Imports of mebendazole were constantly increasing to 625 kilograms in 1974 and 920 kilograms in 1975. It was estimated that domestic production would save about $8,000 per 100 kilograms compared to the imported price. And the excess production could be exported to earn foreign exchange. KIST was also very satisfied that it would prevent brain drain, successfully develop the technology through industry-academia collaboration, and directly contribute to the development of the industry. The mebendazole that was released was evaluated as an excellent new technology in terms of its originality and economy, and was designated as a protected drug. Meanwhile, with the localization of mebendazole, the Korean parasite control business has been able to obtain a stable supply of the drugs needed for mass treatment at a relatively low price.